In the last lecture, we started going over the beginning of the organic molecules, such as carbohydrates and lipids. We went over dehydration synthesis and hydrolysis. So now I'd like to look at proteins. Next one is, now proteins not for energy. We saw that carbohydrates were for quick energy and lipids were for long-term energy, like you use in hibernation or for seed development or for reptile or anything in an egg, right? Egg development. Proteins, on the other hand, are for growth. Okay, so we'll think about definitely for growth and for maintenance. A lot of hormones are made out of protein. Okay, and so these are two major things that proteins are used for. So this is your basic amino acid, and so I'm just going to draw it a little bit down here, right? So I start off with my alpha carbon. I just want to show you how I would go about drawing it, okay? So I start off with my alpha carbon, and then that's the one in the middle. And then I'm going to put my amino group first, because this is an amino acid. And the amino group has nitrogen with 2 hydrogens, right? And then the acid group comes next, and that goes on the right side. And then we put OH. All right, then there's always a hydrogen on the top, and then the R group is going to be the variable group. Hence the name amino acid. All right, the amino group and the acid group. And this is why it's called an amino acid. Okay, and so this looks great. All right, so let me also bring up something else. They come into groups, right? So there's 20 total. And let's keep that in mind. You have 20 total, and they are nonpolar, polar, or electrically charged. There's 9 of them are nonpolar. 6 of them are polar. So this is why we went over nonpolar and polar with you before. And that means that nonpolar, they can't stand water. They fear water. Hydrophobic, polar, hydrophilic, and then the electrically charged would be the remaining 5, right? Where you have the acidic kind, and if they're acidic, they would have a minus charge, or they're basic, and that would have a plus charge. And so there are 2 acidic ones, and there's 3 basic ones, and so that's how that's broken up. All right, this looks great. Also, you might want to learn in AP Bio which ones are aromatic, right? Which ones have benzene rings, and so forth. So now, let's take a look at the different amino acids. There's 20 total. And so in the blue, right, are the R groups that I talked to you about. They were located at the bottom. Alanine, valine, leucine, isoleucine, proline, methionine, phenylalanine, and tryptophan. All those, that's 9 altogether, right? That should be all of our nonpolar. And the green are our polar, right? And then we have our 3 basic and the 2 acidic ones down there. Okay, well, just to keep in mind, now you don't have to know the structures of the amino acids for the SAT2. Just keep in mind that they have that, okay? Number 2, they're going to come together and form what? Peptide bonds. Okay, so let's keep that in mind, that water is going to leave, and a peptide bond is going to form between amino acids. And so let's talk about protein folding. And the primary, so there's going to be 4 levels of protein folding. Primary structure, secondary structure, tertiary structure, and quaternary structure. The primary structure is really, really simple, and basically they've numbered the amino acids for us. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And these are the abbreviations, but this whole thing is about 34 amino acids long. Okay, this is the primary structure, is just what's the sequence? In what order are the amino acids laid out? And remember, there are 20 total, right? There's 20 to choose from, so there's so many different varieties of amino acid sequences that you could probably think of. Okay, there's millions, billions. Secondary structure. The secondary structure is going to include 2 things. One of them is the alpha helix, and the other is the beta pleated sheet. Okay, and then they're going to really just bond every 4th amino acid. The alpha helix is going to take its amino group and bond with the carboxylic acid group, or that acid group, right? And so the same thing happens here with the beta pleated sheet. Every 4th amino acid is going to do that exact same bonding. Okay, go up, down, up, down. 2 kinds of structures that come together. Okay, so after the sequence, they're either going to bend like a beta pleated sheet, or they're going to coil up. And when they coil up, a lot of times that's because of the hydrophobic ends, right? There are some nonpolar residues on the amino acid sequence that want to just hide, and so an alpha helix would be more reasonable. tertiary structure. So the tertiary structure is now your 3D protein. Okay, so by the time we reach over here, we have our 3D protein ready to go. Sometimes it's referred to as a globular protein or a really big protein. That's what you have. You can see some alpha helices here, and you can see some beta pleated sheets as they're pointing out over here. There's a disulfide bridge that brings them together. There's other things that we use besides disulfide bridges. Right? By the way, disulfide bridges are found on cysteine. On all cysteine amino, only the amino acid cysteine can do disulfide bridges. Hydrogen bonding exists, and that's always very big, right? They're big in the secondary, and they're big in the tertiary. Ionic bonding is happening because we have electrically charged amino acids that might click together like magnets. We have van der Waals forces. Nonpolar amino acids tend to come together because of van der Waals forces. Then we have the hydrophobic effect. Same thing, all the nonpolar molecules want to hide inside, the polar ones want to come outside. And so this looks really nice. And then finally, the quaternary structure. This is the fourth level up. So basically, we're going to take tertiary structures and we're going to let those subunits come together. All right, these tertiary structures. So this is hemoglobin. Hemoglobin, you might have heard, is the blood protein, right? Hemo meaning iron, or blood, and globin meaning protein, blood protein or iron protein. That's what these little red discs are over here. These are heme groups, right? They have iron and some more protein attached to it. So there's 4 atoms of iron in this one protein. Now, one hemoglobin molecule, you wouldn't find just one, right? In a red blood cell, that's where they would be found. These little red blood cells, you'd find about 250,000 hemoglobins. And then each hemoglobin can hold 4 oxygens. So one red blood cell can hold 1 million oxygen atoms, and this is huge. Okay, and this is a very big protein. It's one of the proteins we've studied the most. It's because it's so key to our survival. It delivers oxygen, right? This is its job. It lives in red blood cells and it delivers oxygen. And almost every vertebrate has a hemoglobin molecule in its red blood cells. But I wanted to talk about a protein mutation. So what happened here is, this is sickle cell anemia. And I want you to see how the cell, the red blood cell has like a weird shape to it now. It's like a hook, right? Or more like a crescent moon. And rather than being like a flat disc, I usually see red blood cells as, imagine taking a small marshmallow, the one you use in hot cocoa, and you squish it. That's what a red blood cell would look like. This is like a crescent shape, like a moon. This is not what a red blood cell should be like. Now what happened was, there's an amino acid sequence here, but the 6th amino acid was replaced from glutamate. So this is what the original 6th amino acid was. It was a glutamate. And the glutamate was accidentally placed, and a valine is there instead. Okay, this one amino acid change is going to lead to sickle cell anemia. I know that's hard to believe. Really, it's one letter change in the DNA. Okay, so an adenine, instead of being thymine, an adenine had been replaced, which then made the mRNA read GUA, which then made the amino acid valine. The rest of the structure is fine. There's 10,000 amino acids, and one of them gets messed up, and look what you see, you get a whole mutation out of that. Okay, so that's how important the amino acid sequence is, from the primary structure all the way down to the quaternary structure. It's very important. One mistake in the primary structure can lead to mistakes in the secondary, to the tertiary, to the quaternary, eventually into the entire cell. All right. All right, so then protein function. Well, proteins have so many functions. I could never get a list big enough for you guys, but I wanted to point out where they are. They're in cell membranes, right? They're used as transport molecules. They're used in the electron transport chain. They're used in photosynthesis on the thylakoid membrane. Just proteins in the membrane are going to be a very big theme. Enzymes is the second biggest thing we're going to always see, right? They catalyze reactions along the way. They make reactions happen faster. They have an active site. Muscle tissue, myosin and actin filaments coming together. A lot of protein in muscle tissue. Microtubules, right? These are vesicles are going to be transported on microtubules. Chromatids are going to be pulled apart by spindle fiber microtubules. Microfilaments, right? Pseudopods are going to form from the amoeba. There's structural protection, such as our fingernails, right? Our skin has a lot of protein in it. These are structures that help us, or even like antlers on a moose. pigments like chlorophyll, like melanin that we have on us. And finally, what we brought up before, hemoglobin, the thing that carries oxygen in red blood cells. And that's it with proteins. Now the next is the shortest one. Nucleic acid is great for information storage and it's great for energy. And so I put over here a nucleotide because this is the monomer for all nucleic acids. Every nucleotide has a phosphate group, a sugar. Now the sugar is either going to be a deoxyribose, and we brought this up when we did, in the last lecture when we did carbohydrates. Deoxyribose is found only in DNA. Okay, and then there's the other one, ribose. And all the difference is that the deoxyribose is a ribose that's missing oxygen. Okay, it's a ribose that's missing oxygen. And so RNA is used with ribose, but also ATP. And we're going to talk about ATP a little more detail when we start going over cellular respiration and photosynthesis. But right now, just keep in mind there's two sugars, okay, deoxyribose and ribose, found in our nucleotides. Let's take a closer look at the nitrogen bases that we'll see. So we went over this in the one of the first lectures that we had, right? But we went over the purines and the pyrimidines. I hope you guys remembered. Purines, okay, look like this, and they have two rings, right? So there's going to be one hexagon and a pentagon attached to it. That's a purine. And then, of course, they were the pyrimidines. And these guys have one ring. And so the only difference was the purines were adenine and guanine. And then CUT, cytosine, uracil, and thymine for the pyrimidines. Pure as gold, cut the pie. You guys remember that from lecture number 1, and so we don't have to write it twice. All right, guys, and that's it. Nucleic acids being the briefest one of all. We're going to see a lot of this in the genetics unit, and so that's why I kept it very brief for this chapter. Okay, guys, so with that said, make sure you guys keep on reading, right? Watching the lectures, do your practice tests, and like 5 weeks' time, you'll start to see a big difference in your score, reaching up to an 800. Anyway, until next time, I'll see you. Be safe.